This is the solutions video for problem set 13 for Kevin Ahern's BB450/550 course at Oregon State University. In this video, I'll be working problems related to the process of glycolysis. The first question says that glycolysis is unusual in having 3 regulated enzymes. Why is it important that the very last enzyme of the pathway is regulated? Well, let's refresh our memories first about the last enzyme of the pathway. The last enzyme of the pathway says that PEP goes to pyruvate. This reaction is catalyzed by the enzyme pyruvate kinase. Now you remember that this reaction is the reaction I called the Big Bang. And it's called the Big Bang because the delta G0 prime for this reaction is equal to minus 31.4 kilojoules per mole, meaning that at equilibrium, the reaction conditions for this will produce an enormous ratio of product divided by reactant. When cells are doing gluconeogenesis, they must do the following thing. So this is for glycolysis. And in gluconeogenesis, they must reverse the reaction. However, to reverse the reaction, they can't do it in one step. There's enough energy that's released here in this process that it's just too much of a barrier for the cell to overcome to try to simply reverse the reaction. So instead, the cell does what I call the 2-step during gluconeogenesis. In the first step of the process, pyruvate is converted into a molecule called oxaloacetate. That reaction requires ATP, and in the second step of the reaction, oxaloacetate is converted into PEP, and that reaction requires GTP. Now, if pyruvate kinase were not regulated, what would happen is that as soon as the cell went from pyruvate to oxaloacetate to PEP, instead of going to the next intermediate in gluconeogenesis, which happens to be 2-phosphoglycerate, instead the PEP would simply get converted back to pyruvate because pyruvate kinase would be active, and it's so active that all the PEP would essentially go back to pyruvate and virtually none would go into the formation of 2-phosphoglycerate. Consequently, regulation of pyruvate kinase is essential for the process of gluconeogenesis to occur. Question 2 says that a cell has an overly active lactate dehydrogenase. And predict how the cell might behave in low and high oxygen concentrations. Well, let's remember first of all what lactate dehydrogenase does. In muscle cells of animals, pyruvate is converted into lactate by the enzyme lactate dehydrogenase. And this is a reaction that converts NADH into NAD. This process is essential for the process of fermentation. That is, the process that occurs when cells are out of oxygen. And it's necessary because when oxygen is present, NAD is produced by electron transport, and cells have abundant amounts of NAD+. Under low oxygen conditions, however, electron transport is not occurring, and cells need a source of NAD in order to keep their glycolysis reaction going. So lactate dehydrogenase in animals is very important for this process. So now we're asked to predict what would happen to this cell if it had an overly active enzyme and how it would behave in high and low oxygen concentrations. The answer to the question is there would be relatively little impact that would actually happen, because remember that an enzyme speeds the reaching of equilibrium, but it does not significantly change the direction of an equilibrium. It doesn't change the direction. It doesn't change an equilibrium at all. So what we might expect to happen would be under conditions where we have low oxygen, we might more rapidly have the formation of lactate, but we would not expect that we would be tipping the balance in favor of lactate any more than we would under conditions where lactate were essentially limiting. Similarly, under high oxygen concentrations, where this reaction isn't running very much at all, we wouldn't expect to have hardly any effect at all. So the answer is basically we wouldn't expect to see any significant effect for the reactions occurring under either of these conditions with an overly active lactate dehydrogenase. Question 3 says a brewer hopes to speed up the brewing process by adding oxygen to the vat. Predict the outcome of this reaction. Well, remember, of course, that yeast and bacteria have a different process for fermentation than animal muscle cells do. In yeast, we have the following reaction that occurs in the absence of oxygen. Pyruvate goes to acetaldehyde, which goes to ethanol. The first reaction is catalyzed by the enzyme pyruvate dehydrogenase, also called pyruvate decarboxylase. And the second reaction, acetaldehyde going to ethanol, is catalyzed by the enzyme alcohol dehydrogenase. And it's this reaction that converts NADH to NAD+, just like we see in animal muscle cells. Now, if we add oxygen to the mixture in hope to speed things up, we actually will screw things up, because remember that pyruvate has different fates depending upon the cell that it's in and whether or not oxygen is abundant. In aerobic cells like bacteria and yeast, pyruvate in the presence of oxygen, instead of going to form acetaldehyde and ethanol, will instead be oxidized to acetyl-CoA, and acetyl-CoA will go to the citric acid cycle. Consequently, no ethanol will be made. So depending upon how much oxygen was actually added, what would happen is that pyruvate would at the very least be reduced in the amount of ethanol that it would produce, and if sufficient oxygen were added, essentially no ethanol would be produced. The fourth question says that the Big Bang reaction is essentially irreversible. Using the Gibbs free energy equation, calculate the equilibrium ratio of products to reactants for the pyruvate kinase reaction, assuming it is occurring at 300 degrees Kelvin. Well, to do this, for number 4, we must remember a couple of things. First, we remember that the delta G equation says that delta G is equal to delta G, 0 prime, plus RT times the natural log of products over reactants. We remember that delta G at equilibrium is equal to 0. And we also remember that the delta G 0 prime is equal to minus 31.4 kilojoules per mole. We plug in values for R, which are 0.008314, 300 degrees Kelvin, and 0.003814 is in kilojoules per mole a degree Kelvin. This is in 300 degrees Kelvin for the temperature here, times the natural log of the products over the reactants. Okay? Now, rearranging this equation, we get 31.4. I'll leave the units off here, is equal to 0.008314 times 300, okay, times the natural log of products over reactants. So the natural log of products over reactants is equal to 31.4 divided by 0.08314, 300. And if we solve then for the ratio of products to reactants, we say that products over reactants is equal to E to this number. This number turns out to be equal to 12.5. So E to the 12.5 is equal to 267,990 to 1. The ratio of products to reactants, if we run the pyruvate kinase reaction with the delta G 0 prime value that it has, means that the ratio of products to reactants will be enormous compared to the conditions that we might have started with. It's for this reason that cells do the two-step around the pyruvate kinase reaction. They can't simply reverse the reaction effectively in order to do gluconeogenesis, and for this reason they must go through oxaloacetate and then go to form PEP.